All right. Good morning, everyone. Thank you so much for being here. It's another chilly Saturday, and this is a wonderful turnout. We're so happy to have you here. So thank you so much. My name is Heather Robbins. I am the Director of Communications in the Eberly College of Science, and I have the honor and privilege of welcoming you all to the second week of our 2026 Ashtekar Frontiers of Science lecture series. This popular public winter series is now in its 32nd year, which I think is pretty amazing. I can't say this for certain, but I personally don't know of any other lecture series on campus that's been going on that long, and I think it's pretty special. The lecture series was founded by Abai Ashtekar in 1995, soon after he arrived at Penn State as director of a new research center that subsequently evolved to become the Institute for Gravitation in the Cosmos. It owes its success to tireless efforts and meticulous planning by Barbara Kennedy, who presided over the series during its first 25 years, making it one of the most successful science outreach programs in central Pennsylvania. This year's lecture series, titled Unexpected Pairings, addressing today's biggest societal questions using different disciplinary approaches, will explore how researchers in the college approach some of today's biggest societal questions using different approaches. The 2026 lecture series brings together researchers from disparate disciplines to showcase how faculty from across the college's 7 departments leverage wide-ranging methodologies and theoretical approaches to tackle pressing scientific issues and help shape the future of science, health, and humanity. Each of the 6 lectures will feature 2 high-level overview talks focused on one of the big societal questions that drive research in our college. Today and for the next 4 Saturdays, you will hear from our researchers on topics including origins, medicine, resources, materials, climate, and education. And for those, either if you were here last week and you want to watch it again, or if you missed last week's lectures are already online on our website, science.psu.edu/frontiers. And we'll have a link to that later on the presentation as well. Today's first lecture topic, How Computational Modeling is Transforming Alzheimer's Research, is being presented by Wenrui Hao. Wenrui is a professor of mathematics and director of the Center for Mathematical Biology. His research focuses on computational modeling, scientific computing, and mathematical models and artificial intelligence to study diseases such as cardiovascular disease and Alzheimer's. The second lecture topic, How Our Genes Influence the Risk for Neurodevelopmental and Psychiatric Disorders, will be presented by Santhosh Girirajan. Santhosh is a physician by training and a geneticist by practice. He is the T. Ming Chu Professor of Genomics and head of the Department of Biochemistry and Molecular Biology. His research aims to discover and characterize genetic changes that contribute to neurodevelopmental disorders. So here's Santhosh and here's Wenrui. Following the lecture, our wonderful graduate students, I have to say, Katie Yan and Soon Lee, will lead a moderated question and answer session with both of our speakers today. And without further ado, please join me in welcoming Wenrui Hao.
Okay, thank you everyone for coming, especially cold Saturday morning, and also thank Heather's nice introduction and also invitation to give me this opportunity talk about my work in Alzheimer's disease. Okay, so first, simple but important question I want to ask is why it's called Alzheimer's disease? So actually it's named by Dr. Louis Alzheimer, who is a German psychiatrist. And the first patient now we call Alzheimer's disease patient is Auguste Dieter, and got dementia at her age around 51. When she came to see Dr. Alzheimer, and she couldn't speak clearly and cannot answer question appropriate. And this symptom actually is pretty unusual at her age. It's a sampled much older symptom for older patient. Okay, so then at this moment there was no treatment. And then after 5 years, she passed away. Dr. Alzheimer did a post-mortem examination of her brain, and this is the 2 features. So from here you can see there are 2 features. The first one is a plaque, is called amyloid beta plaque. Then you have lots of tangles, is called tau fibrillary tangles. This is still two features we are using to diagnose Alzheimer's disease today. So then people name the disease after him. After first AD patient in 1907, down the road, we can see a lot of milestones for Alzheimer's disease, including discovery of biomarkers, genes related to disease. And in early 2000, we start to collect data, including imaging data, CSF data, plasma data. And we also talk about the treatment and even AD vaccine. But the problem is our understanding for Alzheimer's disease is still limited, and how to prevent and even slow down is unclear. Okay, so what's Alzheimer's disease? So let me show you a picture to illustrate. So this is two curves. One is yellow curve, is normal aging. Another one is blue curve, is Alzheimer's disease. And this horizontal axis is age, and the vertical axis is cognitive function. You can see for normal aging, we have gradual cognitive decline. But Alzheimer's disease starts around age 60 and change dramatically after that. So if you look at the brain, normal brain, you have a kind of fluffy brain, but the AD brain is kind of small and also dry. And look at cross-section, you can see AD start with the memory region. People start forgetting small things, then propagate to language area, so make people hard to speak, and eventually destroy the whole brain. So this Alzheimer's disease is not only theoretical, can affect to real people. So let me show you the one case study. Is one artist, William Utermelon, who diagnosed Alzheimer's disease in 1995. Because he is an artist, he decided, okay, let me use drawing self-portrait to record how the Alzheimer's disease affect him. Okay, so this is after 1 year, 1996, you can already see quite different with what he drew before. Only after 5 years, so this is 2000, this self-portrait is so abstract and that it was very difficult to see himself. So you can see how the Alzheimer's affect cognitive identity and also the artistic expression over the time. And the AD is not only apply this patient, it's also give a huge burden for our society. If you look at the modality, cardiovascular disease is number 1, cancer is number 2, AD is only number 4. But if we look at care cost, these are huge. By this prediction, by 2050, we need to spend more than 1,000 billions on AD care. So this why is the burden is so high? Why is this? What happens in the brain in AD? What's the biology behind it? So let me show you a short video to show what's the Alzheimer's disease, what happens in our brain.
Over a century ago, a German doctor called Alois Alzheimer spotted anomalies in brain sections from a patient with dementia. Ever since, people have been studying the strange plaques and tangles that he saw, in the hope that we could one day understand and cure what is now known as Alzheimer's disease. Plaques are insoluble deposits of a peptide called amyloid beta, or A-beta. They're formed when a protein called amyloid precursor protein is sequentially cleaved by two enzymes, beta and gamma secretase. Other molecules are generated by this cleavage and may play a role in the disease, but A-beta is the main culprit. A-beta tends to misfold and become sticky, eventually clumping together to form soluble oligomers. Some of these aggregate into large insoluble fibrils that deposit in the brain as plaques. Another key feature of Alzheimer's disease is neurodegeneration. Neuronal death and damage is triggered by A-beta, but some of A-beta's effects seem to be mediated by another protein seen in the brains of patients, tau, a component of tangles. In a healthy neuron, molecules are carried along the axon on a series of tracks, made of microtubules and stabilised by tau. But in Alzheimer's disease, tau is modified, causing it to dissociate from the microtubules, adopt an abnormal shape, and move from the axon to the cell body. Like A-beta, tau comes in a variety of forms, and we don't know which ones contribute to the disease. And like A-beta, these forms either remain soluble or stick together and deposit as the tangles that Dr. Alzheimer saw. Eventually, these processes kill the neuron. Despite these advances in our understanding of Alzheimer's disease, no cure exists. While drugs are being developed to target amyloid beta or tau, it's unclear whether they will eventually be successful in treating the disease.
Okay, so from the video, we can see there is no efficient treatment for Alzheimer's disease so far. So why is this? So when we treat the different disease, actually we have normally have a single universal approach to apply for all the patients, for instance, cold or cardiovascular disease. But this is not working for Alzheimer's disease because two AD patients do not follow the same disease path. Because of this, current clinical trial is not working because the patient in the trials could be very different with the patient we want to treat. So in that case, we need to treat patient individually. So the one of the idea is, okay, we can build a digital twin, which is a virtual model to mirror the real patient. Then we can play lots of possible treatment on the digital twin before we treat the real patient. And how do we do this? So we need to use a lot of data. So from the video, you can already learn we have several biomarkers, amyloid beta, tau protein, neuron degeneration. We do need several different sources for each biomarker, for instance, CSF, imaging data, and also plasma. And more than this, we need to track the disease progression. We need to have longitudinal data, for instance, measurement at baseline, 6 months, and 12 months. So this is one of the example have this kind of data in public aiding data set. But this is not only one example. This is kind of many data set can contain this kind of data. Okay, so then let me introduce the framework for digital twin. So the first one, you have a physical twin, which is Alzheimer's patient, and provide a different model data. So we have CSF data, imaging data. Moreover, they have different pathologies. We need to have different causal network for this patient. After that, we can create a digital twin based on this framework. Idea I borrow from multiple agentic AI. So you have different agent. One model is for imaging data, another model for CSF data, and the third model is for causal network discovery data. So eventually, you can build this digital twin. After that, we can give a personalized treatment for this patient. So the whole idea here is we want to test a different treatment on virtual you before giving a real you. So this is the kind of idea for digital twin. So let me walk you through several modules for digital twin. Then I show you a demo how we put them together. And the first one is, can we have a disease progression clock? So this is a clock we want to tell you at which stage you are in disease mitigation, are you in disease acceleration, which stage are you in? So in order to do this, we need to incorporate different data from the public data or from the patient. So this is the data. We have CSF data for amyloid beta and the tau protein, and the neuron degeneration from MRI data, and also have cognitive test score to see what's the cognitive function for this patient. Okay, so mathematically speaking, this is called ordinary differential equation. So X is the different biomarkers, T is over the time. So if we solve this ordinary differential equation, we know which stage are you in. But the problem is we don't know what's the right-hand side because AD is kind of unclear. What's the mechanism is unclear. We cannot build a model based on understanding of AD. But we want to do a data-driven approach. So the idea is we learn the G of X, and the P is a parameter I will talk about later. And we want to learn everything from the whole population. But the challenge over here is different patients have different time scale. For instance, one patient starts AD symptom around 60 and get AD around 70, but another one from 65 got 65, right? How you integrate every patient scale together, so this is what we want, this is what we have. Actually, because I'm a mathematician, here is a quite simple mathematical solution is, okay, let's do a linear transformation, okay? I can introduce a disease progression score, unify them together, and the alpha I tell you when the disease start for this patient, and the beta I tell me how quick you get AD for this patient. So in this case, we can use all the data, we use the population data together to learn the model. Then we identify what's the personalized parameter. Once we have personalized parameter, we have personalized clock to predict each of them. Okay, let me show you the causal network we learned. This is you start with amyloid beta, triggers the tau accumulation, lead neuron degeneration, eventually will result cognitive decline. So this is the so-called amyloid beta cascade hypothesis, which is widely used in clinics. Actually, we learned this one through the data directly. If you like the equation, let me show you the equation. This is the ODE. You have initial condition, then you run with a certain personalized parameter, we can get each patient. So across all the data set, we can see two groups. One is cognitive normal group, another one is the disease group. And of this, we can get around 90% of prediction accuracy for each biomarker, so that we can give a personalized prediction and also disease tracking. So the second question is, can we see how the spread over the brain for each patient for the biomarker? So mathematically speaking, we need a partial differential equation. It's not only time, you also have spatial variable. And we also integrate with advanced machine learning techniques. This is we developed in our group is called Laplacian eigenfunction neural operator. So we can learn the right-hand side. So the idea here is, okay, we fit the PET data, PET and MRI data, then you put all the operator, I can tell you where is the disease starts, how the disease propagate. So let me show you one example on one patient. So this is the, okay, so this is the one patient. We use the two training data for each biomarker. Then we can predict very much later time for each biomarker, even we don't have measurement at all. So if you want to see how the propagate over the time, so this is the amyloid beta accumulation over the time, how you can see it start with very little amyloid beta, then you can propagate to the whole brain. Okay, once we learn the model, we can see how the disease progress over the time, over the brain. So different brain have different regions. So let me show you two patients. Because I cannot show the patient privacy, I just give them Tom and Jerry. You have two names over here. So you can see different color means the different region, and the thickness show how the strength of the connection. Okay, so this is the one patient. You have another patient here. Okay, so this is, you can see is similar, but it's also very different. So that's why personalized prediction or personalized model or digital twin is essential for this prediction. Okay, once we have model, we can give the treatment. So let me give you one example. We are doing is called anti-amyloid beta treatment, for which there are many drugs approved by FDA in this direction. So what we are doing is U of t. So first is the amyloid beta equation. We introduce a dosing function. U of t is how much dose I should give over the time. And the idea here, we want to minimize cognitive decline, C of t, which is a clinical outcome, and also side effect. Mathematically speaking, this is a so-called optimal control problem. So we can solve optimal control problem for each patient. Then we can see what's the efficacy over across the data set in the clinical trial. So here is the conclusion is, okay, if we can give the treatment earlier, around age 60, treat a longer time and a high dose, then you can get a good efficacy around a 5% reduction of the cognitive decline. Okay, as I said, very beginning, every patient, they have different pathophysiology for AD, right? So the question is, how many possible pathophysiologies we have for Alzheimer's disease? We already studied for AD for years. There should be a lot of accumulated knowledge for Alzheimer's disease. Is that possible? We can find all the possible causal network or pathophysiology. So what we are doing is, okay, let's read all the related papers. We look at a highly cited paper from Google Scholar and PubMed and ask large language model, for instance, ChatGPT, to read the paper for us. So we ask the ChatGPT do the following two things. The first one is find the associated biomarker for AD. Second one is tell me what's the causal relationship between biomarkers. So then we can have a causal network. Its nodes is biomarker, age is a relationship between them. So essentially, we can find all the possible network. So this is, I'll give you one example here. So if we look at the recent paper, this is a network is very complicated compared to the 20 years ago, right? So you can see this is the complexity is we do need digital twin to model, reason, and personalize this complexity. Okay, I think I talk a lot on different modules. So your question will be how you put this together, right? How you fit the one patient. So let me show you a demo how this digital twin framework with the different modules, they will work together as a whole.
Our approach is a multi-agent, AI-driven digital twin, a system that doesn't just compute, but learns, reasons, and adapts. In the forward loop, 3 intelligent agents move in unison. The causal network discovery agent extracts Alzheimer's disease mechanisms from highly cited scientific papers using large language models. It identifies the causal networks described across different studies and integrates them into a unified representation, forming a comprehensive causal framework. In this demo, we present the 6 distinct causal networks derived from the literature. Together, they provide the foundational pathophysiological basis for our AD modeling efforts. The CSF biomarker agent is designed to learn from real patient data. We first loaded the CSF dataset and trained the agent to capture the underlying CSF dynamics. Once trained, the agent predicts temporal changes in CSF biomarker levels for each individual. In this demo, we included 5 patients, showing that the agent can generate distinct CSF trajectories for each patient while fitting the provided clinical biomarker data. Next, the imaging data agent transforms brain scans into dynamic, evolving biomarkers. This agent is trained using state-of-the-art neural operator learning to capture the progression of neurodegeneration with high precision. Once trained, it can predict imaging biomarkers for each individual. In this demo, we present 5 patients, showing distinct trajectories for amyloid beta, tau, and neurodegeneration biomarkers. Here, we show our agent's training and prediction results versus clinical imaging data, with over 90% accuracy. These 3 agents form a digital twin that predicts patient-specific disease progression. Next, the backward loop uses the treatment agent. After training, it explores all possible dosing strategies to design the optimal regimen for each patient, identifying the lowest dose, safest schedule, and most effective intervention. In this demo, we show 3 dosing regimens for 5 patients across 3 treatment strategies: an anti-A-beta therapy, an anti-tau therapy, and a combined therapy. This is more than a model, it's a step toward personalized Alzheimer's care.
This is a digital twin framework. As a mathematician, I can see mathematics is essential when we deal with the complex clinical data and make model be reliable, interpretable, and trustworthy. Is not only a black box. Okay, due to the time limit, let me answer 2 questions from mathematical point of view. So the first one is how you integrate the multi-model data. So you have from the different sources, how you integrate them together into a mathematical model. And the second one is how much data is enough. As you know, machine learning is data hungry, and the mathematical model most can make the data use very efficient, but still a question is how much data is enough. Okay, for the first question actually is how you integrate different multi-model data together. For instance, if you take the imaging data, you could take it from the different hospital, different machine have different measurement error. How you integrate them together is essential for digital twin. How do you solve this? Let me share one story from home. So when my daughter was 2 years old, and I asked her, okay, there are 2 eggs, one is raw, one is hard boiled. Can you tell them apart without breaking them? And she didn't think a long time, just go and touch, say, hey, this is warm. So that means she tells the difference based on temperature. So I follow the same idea here is we introduce the temperature concept for each data set. Okay, so then we integrate thermodynamics into data science. Then we can have this so-called entropy enhanced network to put everything into this data. So you can see the T1, T2, T3, T4. This is a different temperature. So I don't go through details, but if you are interested in, I will point out our recent published PNAS paper or newsletter for more details. Okay, second one is more important is how much data is enough. So in another word, if I give you available data, is this efficient to calibrate all the model parameters? So this is the so-called parameter identifiability. So the idea is, okay, let me fit the model to the data, then you test the parameter identifiability for each of them. If you have an identifiable parameter, we can get the trustworthy model. If you have non-identifiable parameter, you get the model uncertainty. So we developed a first efficient computational framework for parameter identifiability from an optimization point of view. So if you are interested, this is our paper in Advanced Science. Okay, so let me summarize what we have. So instead of treating all the patient uniformly, we develop a digital twin framework to treat the patient individually, right? So in this case, you can see how the patient respond for different treatment, and also how you optimize the clinical trial design, and essentially, this should be a faster, efficient, and personalized treatment solution. Okay, looking ahead, I want to include more data. So for instance, genetic data, omics data, and also social and environmental data. For instance, air pollution, social activities, and also the digital health data, for instance, sleeping data or daily activities. Integrate everything, we can have the more complete and the comprehensive digital twin for Alzheimer's disease for each patient. Okay, so let me stop here. I want my students and postdocs, and also collaborators from Penn State and Duke. So this is the one picture we took last year as a digital twin in AD workshop at HUC. And you can see many of people contribute this talk, what I talk about today. So I also thank my funding agency, NSF, NIH, and is Sanofi, which is a pharmaceutical company, want to extend this new digital twin framework to clinical trial design. Okay, let me stop here and give the stage to my colleague and also my neighbor, Santhosh.
Thanks, Wenrui, for the talk. If we can all join me in thanking Wenrui again for his amazing talk. Excellent. Hi, everyone. My name is Katie Yan. I'm the graduate student intern in the Eberly College of Science Office of Communications. I'm also the Barbara McClintock Science Achievement Graduate Fellow and a grad student in biology. As we transition to our next talk, I want to note that all of you, when you entered, should have received a note card from our wonderful Science Lion Pride volunteers. If you want to go ahead and write down any questions you have for our first speaker on that note card, now is the perfect time to do so. While you're writing those questions, I also wanted to share that our math and stats programs in the Eberly College of Science are consistently ranked in the top 10 according to the National Research Council. In addition, this year, Penn State's biology program ranked in the top 10% among biology programs worldwide, according to the QS World University Rankings by subject for again this year. Yes, that's wonderful, isn't it? Alrighty, looks like Santhosh is ready. So if all of you are ready and have your questions written down, we'll move on to our next speaker. Please join me in welcoming Santhosh.
Thank you so much for coming, and it's a pleasure. Thank you so much, Dr. Ashtekar, for supporting this cause, and such a nice venue for us to learn about different types of research that are ongoing here at Penn State. And one of the aspects that I want to reveal is I'm probably one of the only human geneticists on campus. And when you think about genetics, we typically think about similarities. When I say genetics, probably you're thinking about families. You're thinking about people that are similar to you within your family. Who resembles what? It's the same thing for me too. For example, this is a representation of my family as put together by my younger son, three included, I guess. But at the same time, when you think about genetics, you're also thinking about science fiction. You're thinking about how DNA can be manipulated to become a superhuman, something that would give us enormous abilities that we probably did not have without those DNA changes. But in reality, genetics is all about the study of variation. And that variation can be simple human traits about what are the genetic changes contributing to our height differences, the differences in our perception, our visual acuity, for example, our weight, for example, color of the skin, our susceptibility to various diseases, for example, and can also be associated with human conditions. Human conditions that are actually affecting our day-to-day life, our quality of our life. Or if you take this in a really, really long periods of time, how we have come about, who we are and where we come from, can also be all determined by genetics. For example, if you're thinking about changes that happen in our genome for a really long period of time, hundreds of years, can determine what contributes to our height differences. And at the same time, if you're looking for just in a few generations, some new things can happen that can lead to human conditions. And over millions of years of genetic change can lead to evolutionary changes. Changes in the way we look, the changes in the way we perceive the world, and how we actually interact and communicate. So having told you about genetics, I want to tell you about what are the genetic changes we are talking about. The genetic changes can be, well, we have A, T, G, C, about 3 billion bases. Those bases can come from the father and also from the mother. So in total, we have about 6 billion bases of DNA. And those changes can be a change which could be just a single nucleotide change, which is called SNV, or insertion of repeats like GAT, CAG. Some of these repeats can increase in number, as well as you can miss certain locations of DNA in your genome, or you can have certain DNA that can be duplicated. Well, for this crowd, probably you'll understand this one. This is our alma mater song. If you think about this, if you have a small spelling change, a typo, that can be a missense mutation, the way how DNA can be converted into protein can be changed by this particular spelling mistake, right? Then you can also have something that would truncate your protein, leading to a completely different protein, leading to changes in enzyme or structural proteins that make us who we are. All the functions in our body can be changed. Or you could miss a whole word right there that is called deletion, that can remove multiple genes, multiple different functions can go really hampered. And you can also have a duplication of DNA that would actually cause kind of misexpression of proteins that are important for our function, that could lead to completely different types of disorders. Well, how different are we from each other? Like I mentioned, we have about 6 billion bases of DNA, 3 billion bases coming from our father, 3 billion coming from our mother. And studies on about 1 million people have shown that we are actually 99.6% similar to each other. You may think 100%. No. We are 99.6%. And all the differences that we see is accounted by 0.4% of our genomes. Anything that can be a difference in terms of our height, our weight, our cognitive ability, all those things can be accounted by 0.4%. And those genetic changes can be common mutations or common variants, the ones that are very frequent in the population that you and I share. That's 99.6%, right? And those actually arise about hundreds of generations ago. And these are the ones that those changes, the genetic changes, do not affect us much. They do not affect our ability to have children and do not affect the ability of our children to have children. So these mutations are propagated. They are transmitted over hundreds of generations, and this increases in frequency. And these are the mutations or genetic changes that contribute to traits like differences in height, BMI, and also some of the other aspects such as color of our eyes, for example. On the other hand, you can have mutations that are extremely rare, right? Those mutations are actually that are occurring in our immediate generations. And these are the mutations that are very low in frequency, which means that these are seen in very few people. And these mutations occur in every generation, and they occur independent of each other. And they are very private in each one of these families, and they are the ones that are contributing to developmental and psychiatric features. So, in a sense, what we all have are individually rare but collectively common mutations in our genomes. What do I mean by this? Well, when I talk about common mutations, these are mutations we all share. These are the mutations that is common among us. And if we were to count the number of such mutations, those mutations are actually fewer than the private mutations that you all carry. So every family has its own set of private mutations that has been passed on for a few generations. And like I mentioned, more than 90% of the mutations are seen in less than 0.1% of the population. Okay? So we are all carrying our own legacy. We all carry our own private mutations. And those mutations, if you were to think about it, half of those mutations are seen only once. So we are nothing but our own private sets of genomes contributing to who we are. What are the susceptibilities we all have? And how do you really identify these mutations? Well, today you have something called whole genome sequencing methodology. And this would give you a whole readout of your entire genome, all the whole 6 billion bases. And you can find mutations. These cost about $500 per sample to about $2,000, depending on which company you go to, which clinical care you go to. And also I want to remind you, in clinical genetics or clinical care, whole genome sequencing has not been completely incorporated. It's not part of the clinical care. So this is something that might happen in the future because there are multiple different reasons this is not in clinical care. For example, We don't know what mutations contribute to what phenotypes or what clinical features. So this is not yet implemented in clinical care. Well, I talked to you about mutations. I talked to you about how changes in DNA can contribute to variation that we see between us. There's also another reason that we are variable, why we are all different from each other, especially why siblings from the same family actually are very different from each other. There is a small biological phenomenon here. That's something I want to take a step back and explain. During the formation of our sperms and eggs, what happens is the DNA copy from the mother and the DNA copy from the father undergo shuffling. Okay? So I talked about how we get 3 billion base pairs from our father, 3 billion base pairs from our mother. But in sperms, there's only 3 billion base pairs, and the eggs have the 3 billion base pairs. And those 3 billion base pairs are nothing but a shuffle of the DNA coming from both your father and the mother. That shuffling is different for every child that is born. Okay? So I was telling this to my son. I have 2 sons. I told them, if I get you a piece of cake, you get the half, your brother gets the other half. Well, that's how it is in terms of genetic, how you transmit. The half of what you get from your parent and what the other half your sibling gets can actually contribute to different traits, because there's variation already within the genome that you're transmitting to your children. So this is a good example showing how the shuffling happens and how this shuffling.
Due to technical difficulties, 3 minutes of this lecture is missing audio. If you have any questions about the content that was missed, please email the Penn State Eberly College of Science Communications Office at scicom@psu.edu. Again, that is sci-comm@psu.edu. Thank you.
So, okay, this is good. So these deletions or duplications are present in all of us. What does this contribute to? Well, this could be contributing to many traits that we all have been looking at that are very common in the population. What are those traits? Anxiety, depression, idiosyncrasies that we think are kind of regular in the population, which is okay, which would still lead you a regular life, still have lead you to hold a good job, prosper in life, all those type of things. Still, these are contributed by these 10% of mutations that some of us carry. I'm going to tell you what happens to those things. So these are called rare copy number variants because they are less than 0.1% in the population. And they affect multiple genes. They remove or duplicate multiple genes in the population, and they contribute to neurodevelopmental features. In fact, about 15% of individuals with these features are caused due to these deletions and duplications. And those features are autism, schizophrenia, epilepsy, intellectual disability, and so on. These copy number changes can lead to a specific constellation. Well, I have astronomers here and astrophysicists, so I have to use the word specific constellation of clinical features, which means that a physician should be able to see these 5 or 10 different features in a child and say, this is a particular syndrome that I can diagnose. Genetic testing is actually necessary. These are very specific. Every child with deletion and duplication will look exactly the same. They will manifest the same sets of features. That can happen. Or on the other hand, you can have individuals carrying these deletions and duplications, some of them very severely affected, while others are actually apparently very normal. So I'm going to give you one example of those who are actually very severely affected. This is a child who has features of Smith-Magenis syndrome with intellectual disability, craniofacial features, sleep disturbance. They have an inverted circadian rhythm of melatonin. These kids like to be awake during the night, and they'd like to sleep during the day. Okay? And they have an inverted circadian rhythm of melatonin. Every child with this feature has a deletion on chromosome 17, P11.2. What I found out this is like 15, 20 years ago when I was a graduate student, that when you find children having the same features, some of them have a specific size of deletion, while others have longer or shorter deletions of this particular region. But all of them are deleted for one gene called the RAI1 gene, the retinoic acid induced gene. Then what we found is that when you find children with the same features but do not carry the deletions, those children actually end up having mutations in this gene called RAI1. So today in diagnostic lab, once a clinical geneticist finds a child like this, they have only two tests to do. Do they have a deletion of 17p11.2, or do they have a mutation in this gene RAI1? Well, there is also a reciprocal duplication of the same region where the same genes are duplicated. It is called a Potocki-Lupski syndrome. Deletion of the same region is called Smith-Magenis. Duplication of the same region is called Potocki-Lupski syndrome. Children have opposite sets of features. They don't have obesity. They're very lean. They have intellectual disability. And of course, they have autism and growth retardation. And as part of the study, one of the important things to note is, how do you know it's causal? Well, make the mutations in mouse and see if you see the same features. So we reduced the expression of this gene RAI1 in mouse. The mouse showed, which I'm not showing here, craniofacial features. It showed obesity, and it also had hypoactivity. But then you increase the copy of this gene, in mouse, it leads to growth retardation in mice, and it leads to hyperactivity and also abnormal social behavior in mice. So this is a classical example of a syndrome caused due to a deletion, a copy number variant. There are so many such that were discovered after discovery of this Smith-Magenis syndrome region that are associated with these deletions and duplications and syndromes that are very recognizable clinically. After that, just about 18 years ago, the genetic technologies became so affordable that every clinician, a psychiatrist, a child neurodevelopmental physician started testing their children, their patients for changes. They were expecting to find syndromes. They were expecting to find a one-to-one correspondence between genetic change and the clinical features. But again, the complexity of life is that life, I mean, not philosophically, but I'm meaning life like an organism, is that there is no one-to-one correspondence. What was known is that a deletion that we thought is associated with intellectual disability only was later found to be also associated with epilepsy, aggressive behavior, schizophrenia, autism. Similarly, another deletion on chromosome 1q21.1 was associated with multiple different features. And I'm not talking about the same individual having all these features. I'm talking about a psychiatrist studying thousands of individuals finding this deletion. A pediatrician looking at their children with affected children and finding this deletion, same deletion. Essentially, this is a case of the 4 blind men and an elephant, where they're finding, the psychiatrist is thinking, this deletion is associated with schizophrenia. It's something to do with schizophrenia, while the other person is saying, nope, this is associated with autism. So in different families, the same variant can lead to different phenotypes, different clinical features, or even in the same family, 2 siblings are showing different features. So the question is, how does the same mutation cause different disorders? And can we establish a paradigm for complex disease? This would tell us the same phenomenon that my colleague Wenrui talked about. No 2 individuals are the same, and no 2 individuals have the same sets of features in Alzheimer, but I'm talking about neurodevelopment. This is like way early during development. So we identified one particular deletion on chromosome 16p12.1. This is like a genomic address. Where do you live? Where I live right next to Wenrui's house. But the genomic address is location, exactly the location, your zip code is called 12.1. This is a chromosome 16. This is a location 12.1. And this deletion is about 500,000 base pairs. It deletes about 8 important genes. And interestingly, these individuals different individuals. Some of them show intellectual disability, some show developmental delay, some show epilepsy, and some show schizophrenia. So this was discovered way back in 2010. What we found later is the same deletion can be associated with multiple different features, even in normal individuals. When I say normal control individuals, you just randomly sample people from the population, which people did in Iceland. In Iceland, they have an excellent healthcare system where every individual in their population have an electronic medical record. All their DNA is banked. They have genomic sequencing data from every individual in their population. So you walk to Walmart, you walk to the mall, they probably have data on you, who you are, for all you know, in Iceland. But they are using it very carefully. They're using it very carefully. And one of the things that they are trying to find is, what is the normal genomic architecture in our population? They found this particular deletion in C is seen in their population, and individuals carrying this deletion and walking around are actually having cognitive features which is 2 standard deviations lower than the mean. And these individuals don't know what is causing them to really, having issues in their life, but they are carrying this deletion. And they do not actually go to the hospital with any issues, except that they have lower cognitive effect. So what we found, I like to study family genetics because I'm very excited to know why are people from the same family different from each other. That's my focus. So when we studied this particular deletion, we found this deletion is inherited in most cases. I want to contrast this to Smith-Magenis syndrome, where parents are absolutely normal. They don't have any features. The child is the one who carries the deletion. Child is the one who has the features. Parents have no issues. Most of our children are inheriting this deletion from a parent. And the parent, who's apparently normal, like I said, is able to hold a job, able to do well in their professional and personal lives, actually manifest subtle, subclinical psychiatric features such as anxiety, depression, milder learning disability, and so on. And when we found, why is it the child is very severely affected, but the parents are actually apparently normal and having very mild symptoms, we find that the child has multiple rare mutations in addition to the deletion. So what we did was we conducted a large scale study. We recruited about 125 families. These are large families, multiple affected individuals in these families. And I want to say, we did this recruitment from Penn State University Park campus. Through online recruitment, they would send us blood samples to our labs in tubes through FedEx. And then we would isolate their DNA, perform whole genome sequencing, collect all the clinical data through questionnaires, through electronic phenotypic measures. And what we did was we studied families. We also studied people through different ascertainments. How you find mutations, what they manifest, also depends on where do you find them. Where do you find these individuals? Are you finding them through clinics? Are you finding them through population studies? Are you finding them through hospitals? So when we did this, we collected all the clinical features. These are the types of families that we are looking at. This is a typical pedigree, telling father, grandparents, parents, 2 children, and their grandchildren, and the ones with the deletion, what are the different types of clinical features they have. You could tell there are some families where there are more psychiatric features like depression, anxiety, schizophrenia, while there are other families where there are more developmental delay, autism, learning disability, and so on. What did we measure? When we started measuring their features, we found the children have multiple different types of features: behavioral, intellectual disability, congenital anomalies, and so on, as shown here. And the parents show depression, anxiety, sleep trouble, psychosis. I want to remind you, these are parents who did not go to the hospital saying that they have an issue. We actually ascertain them through the children. Children are more severely affected. But when you start conducting tests, giving them tests, the parents with the deletion started showing these features. And in fact, there are also the non-carrier parent, the other parent who did not carry the deletion, also showed some features. They were also showing the clinical features that we actually did not expect. We were only looking for the parent with the deletion to show the phenotype and the child to be more severely affected. So we saw a whole range of features in the child compared to its sibling. The sibling was normal, but the two parents were actually showing the features. So for the interest of time, what I'm going to show you is, I'm going to tell you that when we looked at their genome, in addition to what they have, the deletion, each one of them showed different types of other rare mutation. And the types of rare mutation told us what types of phenotypes that they can manifest. Some of them can manifest schizophrenia features. Some of them can manifest nervous system features. Some of them can manifest congenital or growth and skeletal features based on the other mutations they have. Some of them are short tandem repeats. Some of them are another deletion or duplication. And we were able to do this in these individuals, looking at how the deletion, in addition to the deletion, what else they have and what is the trajectory of their features. The next question is, a couple more slides. The next question is, we wanted to ask, how does the ascertainment matter? Which is basically saying, the clinical features that we are looking at depends on where do you recruit these people from. Like I mentioned, from the clinics versus population versus hospital. So we looked at multiple different types of data and asked the question, the different types of other mutations they have and the phenotypes they have is very variable. If you look at the patterns, the colors are like the more higher or lower. If you see adults, these patterns differ based on where they are, what types of population we're getting them from. Population, which is like a general population from the UK, it's called the UK Biobank, essentially like the Iceland population, and hospital population going to the hospital. They might be going to the hospital for a completely different reason, but we are finding them with the deletion and what features they manifest. And the ones which are from completely, this is the all of us, which is the population level data from the US. This is called the all of us data. And compared to them with children with developmental delay, children with autism. One quick note I want to say is, children with autism are less likely to have another mutation elsewhere in the genome. When they already have the deletion, they manifest autism. That is it. So they have fewer other mutations. That is why you see blue here. The whole thing is blue because there are few genome compared to the developmental delay children. Well, if you put them all together, we have a lot of numbers, what we call a statistical power to find some associations. We're able to find which other mutations contribute to what trajectories. So the next question that you might ask, which is something which we wanted to know, how are these mutations actually coming together? The deletion is coming from the one parent, and the other parent is actually affected too. Where are these second mutations coming from? The question that we want to ask is family history. So what I'm showing you here is the some of these phenotypes in the child, male versus female. It's ADHD, anxiety, bipolar, depression, OCD, and so on. What we are finding is, in this light green, neither parent have a family history, which means they are not affected at all. They're perfectly okay. The child is less likely to show any phenotypes. When both parents are having a phenotype, having some clinical features, the children are more likely to show features. So what does this essentially tell us? It tells us that we started looking at both parents' clinical features, and we find the scores, social responsiveness score, a score for autism, is actually very correlative between the father and the mother. In fact, this is a phenomenon called associative mating, where tall people tend to find partners who are tall. Similarly, if people with a certain IQ scores tend to find partners who have similar IQ scores. It's also socioeconomic status. Well, my favorite example is, like, if you're a heavy metal rock fan, you're going to find another metalhead as your partner, right? So this is exactly how this is happening, where we are finding some of the features that we're finding in parents actually can predict the features in the child more than the genetic features. So this is exactly what we are finding here. The features in the parents are contributing to the features in the children. And what is the phenomenon? The phenomenon here is, Over generations, because of associative mating, one parent with the deletion passes on the deletion. The other parent, who is also similarly affected, carries other mutations. And these mutations come to the child, and over generation, this gets compounded. The mutations accumulate over generations, leading to a situation where the child is very severely affected and is not able to have more children going forward. So this is exactly how new mutations arise in the population, and those mutations, where do they go? They end up being more severely affected down the generation. And the example here is the 16p12.1 deletion. So final slide, just to show you, this is a population and how features arise in the population. You see some of these mutations occurring in the population, what we call as a healthier cohort. Then these cohorts can get into the healthcare cohort, which means some of this can manifest as something that you go to the hospital. And if it is very severe, these are the children that you see that are the ones that actually end up going to neurodevelopmental clinics. So these are arising here. Some of them are subtle, subclinical, that you really don't see them in hospitals. And as they accumulate more and more mutations every generation, they end up going to the hospital, and some of them going to more neurodevelopmental clinics. So this is work from a lot of students in my lab. I've had about 80 undergraduate students in the past 14 years I have been. And each one of these students have stayed for at least three years in my lab, and my graduate students who were part of this work. Thank you so much again. And this is an wonderful opportunity for me to present my work. I really appreciate it. Thank you.
Hello, my name is Sun Lee, who will moderate the question and answer section. I'll start some simple questions to start our talk, question and answer. So today's themes are like unexpected pairings. So I was curious about, do you guys see any future like collaborations? So I do remember in the math, they need a lot of more data. Do you think like genome can also include it in the data? And the other way, can digital twins can like predict the disease?
Yeah, so maybe I'll answer first. So exactly, this is the my slides I already mentioned is during the future work is genetic data is one of the future, right? So you can include the ApoE4 and PNSI is kind of the genetic data for Alzheimer's disease. I also want to ask Santhosh, how do you think?
I want to say there's only so many base pairs in the human genome, which means there are not infinite number of possibilities. There are actually finite number of possibilities. It is a lot of possibilities. It's probably millions, billions, maybe trillions, but still it's not infinite, which means if you have a lot of GPU power, we can actually model all of this. And I'm also of the belief, well, scientists, when we get into a specific focus, we also become believers. So I should say I'm a believer that, which is true, everything is genetic. You know, everything has a genetic predisposition. Even accidents that happen, the person who's getting into accidents has a genetic predisposition. Whether it's high or low is a different question. Everything has a genetic predisposition. So what I'm going to say is the modeling is what actually the biology needs, because what we are now at a point is generating the large number of data, and our genomes, that my clinical colleagues, me wearing that physician hat, which I used to be in my past life, is that whenever I talk to people saying, oh, genomes, they say, well, clinical features are more important than the genomes. You have to know, have you tested everything that you know? So that's another aspect about Alzheimer's disease. There's also this might, the loop might be complete where there are probably common features that could be contributing to neurodevelopment and neurodegeneration. It is possible that we might already know when a child is born, the susceptibility to Alzheimer, Parkinson, ALS is possible to detect because it is hidden in our genomes. What is going to happen later on is also a matter of figuring out what could be the other risk factors. So absolutely.
Thanks for your answer. The next question is for Dr. Hao. Is the digital twin method already used in a treatment of Alzheimer's disease, or is still on the research level?
So right now, this is still people talking about a digital twin for Alzheimer's disease. Before, it's talk about a personalized medicine or precision medicine. It's already used in cancer or cardiovascular disease. Is for Alzheimer's disease is still the research wide. But as you can see, so for instance, we got a Sanofi partnership with them. We had a meeting with them yesterday, and they want to push a new clinical trial. So there is the two of them. One is the TRIM2, another one is the TRIM2 is kind of, they target several of these, amyloid beta, tau protein, and the neural inflammation. And how they put the treatment over there, that's why we got a grant from them. They want to really push this to the reality so we can see how the digital twin framework can give them what's the best dosing function. So this is not only the research, yeah, more and more attention from industry and the pharmaceutical company and their clinics.
I see. Very thank you. And also one more question for Professor Hao. Are digital twins used also for other diseases, and which diseases are best suited for such individual treatments?
Right now, I think the pretty successful one is cancer, right? So people always talk about personalized medicine for cancer or target, right? Target treatment. So it's already pretty successful in cancer. Is in terms of it's called precision medicine of digital personalized medicine. But for digital twin, people can see is like Alzheimer's disease is not only a disease, it's a syndrome, right? It's not only the genetic, it's kind of your lifestyle, right? So how the lifestyle eventually contribute for a later stage. So that's why is a digital twin is you can integrate all kinds of possible factors together, right? Like Santhosh said, it's genetics, gene is everywhere, but the problem I think is more is the probability, right? Which one you go with, yeah.
And next question is for Dr. Girirajan. You spoke about the correlation between genetics and variation development issues. What about substance abuse? Does it have a genetic components?
Substance abuse? Yes. So it's very nicely put by Dr. Hao. These are all what we call as effect sizes, which is basically the probability of having the disease given the mutation. Substance abuse. So some of these are 100% genetic. For example, Smith-Magenis syndrome, the child has a mutation in REI1, the child will manifest a feature, unfortunately, will show the features. And very rarely we are finding, this is because you look at the large population, some individuals who may have what we call a Mendelian disorder where you have the mutation, you should show a phenotype. Very, very rarely you see somebody who do not show the feature. Those are rare, precious people. We want to know what is protecting them. Similarly, substance abuse is one of those where there's a genetic predisposition. Like I mentioned about a person having predisposition to get into accidents, where if you are careful, well, you can probably not get into accidents. That might be a perception of visual acuity, your hand-eye coordination, whatnot. Substance abuse is also one of those where the effect size is not as high as Smith-Magenis syndrome, but there is a genetic component. And if, for example, this is one of the things that we do in developmental genetics. Once a child is identified to have a mutation, immediately they start speech therapy. They ensure that there's some kind of a rehabilitation. The child then does better than what is expected out of that mutation. Similarly, if this can be done in a way that you identify certain predisposing factors, genetic factors, of course, we can take steps early on. There's a huge genetic component. There's a huge environmental component for many of these disorders. So absolutely, there are many disorders that are 100% genetic. Many of them are actually genetics plus environment, like diabetes. There's a huge environmental component, but there's a huge genetics component too. So it's a 50% genetic, 50% environment. But the 100% is like we have to figure out which part that you want to. The environmental part is easy to manipulate.
Thank you so much. And the next question is also Dr. Girirajan. How does genetic info help with interventions, like medical or educational?
Oh, yes. So this is a huge part. One is The easiest part is knowing which mutations contribute to what features. And that is actually the first part of what we call the genomics era. Now we are in the post-genomics era, where we are facing that the features that we are seeing in the population are amongst us. This is affecting us, but we don't know. And now finding what it is that's affecting us, first. Second, finding what mutations that are. Then once we know what the cause and the effect, then the next step is rehabilitation. The ones that have been that has been possible is like common disorders like hypertension, diabetes. We know the genetic predisposition. We know what is contributing. Then we can take measures for interventions. Similarly, for children, speech therapy, therapies that we can actually identify them early on. So many a times, autism, many children don't get diagnosed with autism unless they're later, like 6 or 7 years. By the time their early development is already done. So it becomes very difficult to really go through rehabilitation. So yes, interventions is a key. Early diagnosis is the key. So genetic diagnosis is an important thing before we can get into intervention.
The next question is, Dr. Can you run the Alzheimer digital twins backward in time to identify the markers of the disease so that people could start treatment?
Yeah, that's a very good question. So normally we did this is we treat the initial condition as an unknown. Then you find the kind of parameter fitting to run it to identify it. But I'm a mathematician, right? So it's not a wild post problem. If you solve from here, you solve this ordinary differential equation or partial differential equation is wild post forwardly, right? If you solve the backwardly, it's not a wild post problem. So then not wild post problem, that means you could have multiple solutions. It's not the only solution is unique. If you solve the unique forward is a unique solution, it's backward, you could have multiple solutions to get this solution. So in that case, it's how many things we can trust the model, right? So if you want to develop a trustworthy model, is at least mathematically speaking, we should have theoretical guarantees. So this is one of the challenge we have, but we can have multiple. It's not the many of this. It's could if you can identify some of possible initial conditions, then we can see which one is the most close one. Is one of the solutions we could do that. So mathematically speaking, this is called, okay, I put a regularization, right? So regularization means, okay, I tell you it's maybe close to which solution, it's not go far away. So it's near this solution I can solve. Okay.
So much. And the next question is for Dr. Girirajan. Does age of parents influence the generic shuffling at time of concession?
Oh, age of parents, very important question. Well, what we know is that from 1935, this is our physicist colleagues might be interested to know. One of the geneticists, I want to say this is my person from my tribe, is a person by name Lionel Penrose. Roger Penrose's father identified that age of the mothers is associated with risk for Down syndrome in 1935. He published a paper in a journal called Journal of Genetics. It's an Indian journal. Way back when that was published way back, where they found this is increase the risk, which has actually led to diagnosis when the age of the mother is over 35. There's a prenatal testing which has been mandated, and the risk also increases. So this was known for a long time, now close to 100 years, like 90 years. But what was not known, which I don't know if I should say, I was proud to be part of this work way back in 2010 that we published a paper, is that age of the father increases the risk for autism and schizophrenia. Why? Because we were able to even draw a mathematical regression plot to find around 42 years, the risk doubles. The age of the father being 42. Why is this? Because the number of mutations that happen in the sperm increases. it increases drastically. On the other hand, if you were to ask me about mothers, why the age of the mother, there's a phenomenon called non-disjunction, which means during the fall, like I mentioned, a lot of things happen that is all developmental, happens during the formation of sperms and eggs in our bodies. During the formation of eggs, the chromosomes have to separate because we are all, the egg is a haploid, which is only contains half copy of a genome. Sperm should contain only half copy of our genome. The chromosomes should separate. But when the age is older, in older mothers, in the formation of eggs, the chromosomes become sticky. And chromosome 21, chromosome 16, chromosome 18, those chromosomes end up more sticky. And then the child ends up having extra chromosome. That is where you have Down syndrome. And also I want to mention the most common cause for trisomy, which is more three chromosomes sticking together, is not chromosome 21, that's Down syndrome. It's chromosome 16 that I study. But you never see those children because those children don't make it. And age of the father actually contributes to autism and schizophrenia. This is which is now well established and validated.
Thanks for answering.
It's a very good question.
The next question is for both. Is there enough human genetic data to use Dr. Zhao's mathematical model?
I want to make a note before I say this. I've had, like I mentioned, I've had 80 undergraduate students in my lab. Many of them come to my lab because they want to go to medical school. I tell them, this is an example now I have because, Wenrui is my neighbor. I did not know that he was, this is the same Wenrui that I see, in papers. And so this is such a nice opportunity. And I see him, he says his child is now 5 years old. I've always seen him pushing the stroller in my neighborhood. And I just wave with my hand. He waves his hand. That's it. I used to think he's in chemical engineering, but I realized he's in mathematics. I want to tell my students, you can contribute to a better health by being a mathematician. You don't have to be pre-med to be a good doctor or a good scientist contributing to health. This is very important. And I tell, our colleague Paul Milewski that I'm reading about Fermat's last year, or I'm reading about John Conway's biography. And he's like, I'm fascinated by how patterns arise in the population in biology. So for me, I think the biggest question, the biggest fascination I've been having is, of course, treating children with autism, but the patterns in which these mutations come together and how it contributes to certain outcomes, not necessarily something that is disease, but traits. How can somebody be taller than others? The genetics is actually showing all those beautiful things. I'm sure astrophysicists and astronomers would be really appreciative of this. These are the patterns that we see in genetics. And we have close to about 1 million individuals' genetic data that has all types of phenotypic information. And the programs such as the All of Us, the UK Biobank, all of them are actually actively recruiting just people from the general population, asking them to give us every possible data, including DNA, blood samples, so that can be registered for research. And what do? Naive geneticists who are kind of wannabe mathematicians do simple regression, some of the machine learning models, without getting into sophistication of mathematics. We need Wenrui if you want to treat we find genetic causes for disease. And absolutely we need more analytical methods, models, and it cannot be done just by biologists or geneticists. So I think this is something to think about.
Thank you very much. I really appreciate our input for high speak of mathematician. So actually we had a meeting 2 days ago with all the Alzheimer's disease research at Penn State, a small group. People always talk about, okay, we have beautiful mathematician, modeler, and also engineering who are working on Alzheimer's disease. But the most fundamental question is from biology. So you need to tell us what's the problem we should do. Then we can figure out what the details, then for the model, everything. So how you do it, the purpose is very important, right? So I think that everything is, we should work together at least, at the most, especially for the complicated disease, for instance, Alzheimer's disease or cancer, right? Whatever disease, is not only one factor. Genetics is very important, the lifestyle is important, biomarkers discovery is important. one of the things we can contribute one of the each, then essentially we can move this whole world forward. I think that's quite important. But I really appreciate how you think mathematics is not only use pen and paper, right, solve the equations, we have some usefulness. Okay, thank you.
No, that's the real science. I absolutely agree. That's a fascination. I want to just add, my foray into medicine was just because I wanted to please my mother. No, I'm Indian, so I have to be either an engineer or a doctor. My brother became an engineer. I wanted to be a scientist. I actually wanted to be a physicist. And well, I had to please my mother. I'm a doctor. Even today, she's not happy that I'm a scientist. She says, "What are you working with mouse and rats and stuff, mice and rats?" And so, well, but my father was the one who actually said, "Oh, you can." So I finished my medical school, and then I went to get my PhD. So so this I think the real science is math and physics. This is where it is. So I absolutely agree with you.